The data we collect from the sample are collected after the researcher has stated the hypotheses and established the criteria for a decision. This sequence of events helps ensure that a researcher makes an honest, objective evaluation of the data and does not tamper with the decision criteria after the experimental outcome is known. Next, the raw data from the sample are summarized with the appropriate statistics. For example, the researcher would compute the sample mean. Now it's possible for the researcher to compare the sample mean, the data, with the null hypothesis. This is the heart of the hypothesis test, comparing the data with the hypothesis. We will accomplish this comparison by computing a z-score that describes exactly where the sample mean is located relative to the hypothesized population mean from the null hypothesis. In step 2, we selected criteria that will allow us to compare a z-score we compute from our sample data mean with the population mean. Now we will calculate the z-score that identifies where our sample mean is located in this hypothesized distribution. The z-score formula for the sample mean is on the slide. In the formula, the value of the sample mean is obtained from the sample data, and the value of the population mean is obtained from the null hypothesis. Thus, the z-score formula can be expressed in words as outlined on the slide. Notice that the top of the z-score formula measures how much difference there is between the data and the hypothesis. The bottom of the formula measures the standard distance that ought to exist between the sample mean and the population mean.